with Jharni. So, in that video tutorial, I am going to discuss with you about HIV virus. Okay, so I am going to wrap up all the things that you need to know from a basic introduction of HIV, then the structure of HIV virus, all from GP120 up to the two single-stranded identical RNA genomes, positive sense. And then we will go to discuss about the replication machinery and then the inhibitors that are present to block the viral replication cycle and how the virus released from the infected cell by snatching a amount of cell membrane from the host cell. Okay, that means this video is going to be a total discussion of AIDS, okay, or more specifically HIV virus. So let's start. So first of all, the thing that you need to know is HIV virus is the causative agent of AIDS, that is acquired immunodeficiency syndrome, although it is very common to us. So there is a point that is quite confusing among peoples, that after infection of AIDS, infection of HIV, that means when a person gets infected with HIV, that does not mean that person will develop AIDS immediately. It takes near about 7 to 8 years to develop the AIDS. Okay, that means suppose if I am infected with HIV, that does not mean I will immediately develop AIDS. It takes 7 to 8 years to develop AIDS. Okay. So what are the symptoms of AIDS? So AIDS, the main thing is AIDS destroys immune system. Why? Remember one thing, that HIV usually loves to interact with CD4 molecules. Okay, and we all know that CD4 molecules are important players of immune system. If they become infected and eventually destroyed by HIV cell, then how our immune system becomes stable, right? So that is how HIV destroys the immune system. And severe impairment of the immune system leads to opportunistic infection, some neurological disorders, secondary neoplasias. Obviously, because when our immune system is compromised, remember one thing, that there are various important normal flora that are present in our body that are usually opportunistic, right? That means they usually do not cause any kind of problem as our immune system is there, right? But whenever due to such kind of problems, such as one of the problem is AIDS that compromise the immune system, they become active, and as they observe that they have now the chance to cause infection. One example of this is esophagus candidiasis. Okay, so esophagus candidiasis are mainly observed in those people that have already developed AIDS, right? And although our candida albicans is usually a normal flora of our human body, right, that we have discussed in our fungal lecture video. So despite being normal flora, as they are opportunistic and HIV provides the opportunity to them, that is why they have caused the infection. Simple. Now this HIV virus is belong to the group of retrovirus. Okay, so now what does this retro means? So remember one thing, that in normal cases, what is our central dogma? In normal cases, our central dogma is DNA. From DNA we will have RNA via transcription, and via translation we will have our protein, right? But in that case it is different. Despite being the DNA as the genetic material, here RNA is the genetic material for that virus, which is also quite common, right? Because we have another RNA viruses that possess RNA as their genetic material. But here the interesting fact is, by you taking RNA as their genetic material, they directly does not go for protein translation, right? But instead they go for a reverse transcription and produces DNA, which is usually known as complementary DNA, right? So, and this DNA will ultimately gives you viral mRNA, genomic RNA by transcription, and viral mRNA will give you viral protein, and viral protein along with genomic RNA leads to the formation of new infectious HIV virus, right? So that is why, as they are performing a reverse thing in their central dogma. That is the reason why they are belong to the group of retrovirus. Okay, retro means reverse. And another important thing that you should remember from that central dogma is, as they are going to perform a unusual thing that are not usually observed in case of host. That is why they carries their reverse transcriptase. Okay, they have to carry this because they do not have reverse transcriptase inside the host, because host does not have any use of recombinant reverse transcriptase, right? And that is why they do not incorporate them. So for their own purpose, HIV have to carry reverse transcriptase with their own. Okay, so that is the quite discussion of central dogma, although we are going to discuss about the replication cycle in details within a minute. So there are some statistics that you need to remember if you want. 32 to 35 million people every year or now in earth are carrying AIDS. 2.5 billion cases every year and 2 million deaths every year observed. In Africa, among them, Africa is the most predominant place where AIDS occurred, along with Asia and some other countries. Okay, so now let's discuss about the classification of HIV virus. So HIV virus is classified onto two groups. One is HIV-1 and HIV-2. HIV-1 are highly transmissible and are predominating in all over the world, right? Whereas HIV-2 is less transmissible and only confined in Western Africa. Okay, and remember one thing, that this HIV-1 and HIV-2 possess only 40% homology. That is the thing you need to remember. And another important thing, that HIV-1 on the basis of the protein that they possess, that is GP120. I am going to discuss about what is GP120 in very much details, but for now let's remember that GP120 is this. GP120 possesses a antigenic variation site, that is hyper variable region. Okay, and depending on their variation, HIV-1 can be divided into another three types. One is clade B. This clade B is predominant among homosexuals. Another one is clade E. This clade E loves to bind with female genital tract. Another one is clade C, and this clade C is unfortunately predominant in India. So clade B is found in Western Europe and America, and this clade B loves to interact with macrophages. Okay, clade E, I have told you that female genital tract, and clade C is predominant in India. So you have to just remember that HIV-1 can be further divided into three classes that are commonly known as clades. Okay, and this separation is on the basis of their differences in GP120. Okay, or more specifically the antigenic site of GP120. Okay, so let's move. Now we are going to enter in our most important part, that is the HIV structure. Okay, so although I have provided a note here, but I am going to discuss this with you diagrammatically. So we are going to discuss about HIV virus from outside to inside. Okay, so there is a lipid bilayer that is found outside the HIV. Okay, see, now this lipid bilayer is not their own property. Okay, this lipid bilayer was snatched by HIV virus during its release from a host cell. Okay, that means it is host property. Now in that lipid bilayer you will found a transmembrane protein present. Okay, and above that transmembrane protein there is a cap like structure present, that is GP120. Okay, and this transmembrane protein is known as GP41. Okay, so that's I have drawn here. This cap like structure is GP120 and this on GP41. Okay, and this is the lipid bilayer that usually present outside the HIV structure, and that is why this virus is also known as enveloped virus. Okay, and this lipid bilayer is coming from what? This lipid bilayer is coming from host cell membrane. Okay, that is stolen from the host cell when they are exiting from the host cell via padding. Okay, now let's discuss about this GP120 and GP41 stuff. So if I want to zoom this picture, yes. So the first interaction between the GP120 and CD4 containing molecule is known as primary attachment. Okay. So now before going to discuss about primary attachment, we should know that what are the CD4 molecules. Okay. So first CD4 molecules are obviously our T helper cell. Not only that, T helper cell monocytes are also helper T CD4 containing molecule. Macrophage also CD4 containing molecule. Microglial cell, follicular dendritic cell, Langerhans cells are all CD4 containing molecules. Okay, and among them except TH cell, that is our helper T cell, all possess CCR5 as their chemokine receptor. Okay, and only helper T cell possess CXCR4 as their chemokine receptor. What is the role of chemokine receptors here? I am going to discuss. So that is the idea. So the primary attachment is between GP120 and CD4, right? Once this primary attachment is accomplished successfully, the GP120 undergo some conformational changes, and these conformational changes will expose other active site present on the GP120. Okay, and this GP120's conformational changes will expose active site. This active site is going to react with the chemokine receptors. Okay, I am again repeating that after computation of the primary attachment, okay, that is between GP120 and CD4, there is a conformational changes going on in the GP120 that will expose a active site. Okay, and this active site will going to react with CCR5 or CXCR4. Okay, that is the secondary attachment. Although here you need to remember that whenever a HIV virus naturally first infect a host cell, their first preferable choice is CCR5 containing CD4 molecule. Okay, then with time they will go for CXCR4, that means helper T cell. That means macrophage and stuffs are primary choice for HIV to infect, right? They do not go into infect helper T cell at first. Okay, so scientists start thinking about if they can develop a drug to block CCR5 for GP120's secondary attachment. Okay, so as they successful, yes, obviously they get success here, and the drug that we have here is Maraviroc. Okay, that is you need to remember. So this is a kind of attachment inhibitor, right, that inhibit the interaction between GP120's active site along with CCR5. Okay, now there is another information that I want to give you, that in Northern Europe and descendant white people, okay, that are near about 1%. Okay, this 1% people are having homozygous mutation in CCR5. Okay, again I am repeating that in this one, Northern Europe and white European descendant white people, okay, having homozygous mutation in CCR5. So what is the utility of this having homozygous mutation of CCR5? Because in that time the CCR5 cannot interact with secondary interaction. Okay, so what is the interpretation of that? So interpretation is these Northern European and descendant white people that are usually 1% of the total are naturally immune to HIV virus. Okay, that means HIV cannot enter to their cell. as they lacks or they have mutated HIV CCR5 chemokine receptor. Not only that, among 10 to 15% people also possess a heterozygous mutation in CCR5. So, what is the utility of this? Here the HIV and subsequently AIDS will spread more slowly rather than normal. Okay. So, usually after HIV infection it will take near about 7 to 8 years to develop AIDS. In that case it will take more. So, that is the idea. So, we have discussed that in case of CCR5 we have developed a inhibitor, that is maraviroc, and CCR5 is also naturally immune, right, and this immunity is due to homozygous mutation in CCR5 along with heterozygous mutation, right. So, after the GP120 interaction, now there is another idea that is going on in scientists' mind, that is, can we develop a vaccine against GP120? That means, can we develop or can we induce our immune system to produce an antibody that will recognize the GP120? If we can, then this antibody will neutralize the virus, and then there will be no longer problematic problem or about AIDS. So, is it possible to develop a vaccine for GP120? The answer is no. Why? Because remember, in GP120 there is a position that is actually antigenic or that will induce the immune system is a hyper variable region. Okay, and in this hyper variable region they continuously undergo antigenic variation. So, what kind of this antigenic variation is? Suppose you have made a vaccine against GP120 in which the antigenic portion that will induce your immune system was star. Okay, so your immune system has learned to make an antibody against the star. Okay, memory cell incorporated it. Now it will again enter in the cell in the form of moon. Okay, then how can the star antibody that was developed can recognize this one? That is the idea why we cannot make any kind of vaccine against GP120, because GP120 constantly changing itself, right. So, we cannot develop vaccines. So, instead we have to focus on creating drugs. Okay, so that we are creating. We are going to discuss about various kind of drugs that are usually used in GP120. Okay, so after discussing about GP120 lot, and I have also discussed about the on the basis of the changes in that GP120, HIV1 is further divided into clade B, come on with me, clade E and clade C, right. Now, after GP120 we are going to discuss about this transmembrane protein, right. This transmembrane protein is GP41. So, what is the utility of GP41? After the successful interaction with primary and secondary successful interaction, this transmembrane protein will hang the host cell membrane and pull towards the virus. Okay, that means it facilitated the fusion. So, can we prevent this fusion of by GP41 of the host cell membrane with the virus? Yes, we can. How? There is a drug named enfuvirtide. Do not bother about spelling because you will get all the example of drug here. I have incorporated for you. Okay, so let's see here. This is your enfuvirtide. Okay, so you don't need to bother about that. Just focus on the concept. So this is the enfuvirtide, that is the GP41 inhibitor that will inhibit the fusion. Okay. So after that, let's enter inside the virus. So after discussion of lipid layer, we want to focus on the matrix that are inside. Okay, This matrix is produced by a matrix protein named P17. Just remember P17 for now. We are going to enter in details too. And inside the matrix we have capsid. Remember one thing, this capsid have 20 faces or side, and on the basis of this capsid is known as icosahedron capsid. Okay, and this capsid is made up of a protein known as P24. Now let's focus on what is present inside the capsid. Inside the capsid, virus has two identical positive sense single-stranded RNA molecule. You have to keep in your mind that these RNA molecules are not complementary to each other, they are identical. Okay, and another thing, what is positive sense? Positive sense means these RNA have the ability to translate into protein. Okay, so that is why positive sense. Along with these two positive sense RNA molecules, there are three enzymes that is present. One is reverse transcriptase, second one is integrase, and third one is protease. And this is another kind of nucleocapsid protein present, that is P7. So that are the various kind of protein that is present inside the virus. Okay, so first of all, the lipid bilayer envelope snatched from the host cell membrane, and this is the matrix protein, and this matrix is obviously virus own property, and this is the capsid that we have, and this capsid is made up of P24. Okay, and inside the capsid we have two single-stranded identical positive sense RNA genome. Okay, and this RNA genome is their genetic material, and we have three enzymes: reverse transcriptase, integrase, and protease, and we have nucleocapsid protein, that is P7. Okay, so let's move to the next slide. Okay, so now we can enter directly in our diagram. Okay, so let me take a color. So this is the primary attachment between the GP120 and CD4 going on. After the primary attachment, this is the secondary attachment with the another active site of GP120 with the chemokine receptor, that is CCR5 and CXCR4. Here CCR5 is the default choice and CXCR4 is the choice for later. And we have here a inhibitor that we can use, that is maraviroc. After that, the third step will be fusion, that is facilitated by GP41, and we can also inhibit this with the help of enfuvirtide. So fusion is going on, and after fusion our capsid containing particle is inside the host cell, as you can see. After that, this capsid will be degraded by host cell protease. Okay, so all the genetic material, proteins, enzymes that are present inside will be exposed to the cytoplasm. Okay, and after this exposition, first enzyme that will activate is reverse transcriptase, and this reverse transcriptase will have two activities. One is our polymerization activity, another one is RNase H activity. How they going on, I am going to discuss. So this is the two positive sense single-stranded RNA virus, right? So polymerization activity of reverse transcriptase will give you a RNA DNA hybrid. Here I am just using one RNA, or in natural it is two. Okay, so this RNA DNA hybrid, now the idea is this polymerization activity of the reverse transcriptase can be blocked. Okay, so this blocking can be done with the help of using nucleotide reverse transcriptase inhibitor. What are they? They are nothing but pseudo nucleotides, right? Because remember, this reverse transcriptase polymerase use the DNTPs that are present inside our cytoplasm as their building blocks and prepare the RNA DNA hybrid, right? If let's remember what we have discussed in case of Sanger sequencing method. what here in that time we are using dideoxynucleotide, right? What are dideoxynucleotide? Dideoxynucleotide means nucleotides that lacks 3' OH group. Okay, and if it lacks 3' OH group, it cannot interact with the next 5' phosphate group present in the next nucleotide. So what will happen? So it result in the simple termination, right? So here we are going to use the same technique. So here the lamivudine, didanosine and zidovudine that I have mentioned as a nucleotide reverse transcriptase inhibitor are nothing but pseudonucleotide molecule that lacks 3' OH group. Okay, and as without 3' OH group, even our host DNA polymerase cannot work, right? Because we have discussed that host DNA polymerase require primer that will give it a 3' OH group, right? So if host DNA polymerase cannot done, virus also not, right? So that is why we can use lamivudine, didanosine, zidovudine, but it poses some side effect. Why? Because remember, we are incorporating pseudonucleotides in the cytoplasm, right? So now instead of using pseudonucleotide by our reverse transcriptase, this nucleotide can be used by our host cell DNA polymerase too, and if it is going on, then it can cause dangerous problem, right? Because it will inhibit the simple replication that we need. So one of the side effect that is going on in such case is bone marrow failure. Okay, so instead of going for nucleotide reverse transcriptase inhibitor, scientists now have developed nucleotide reverse transcriptase inhibitor. An example is nevirapine. Okay, so what they do, they will, instead of incorporating pseudonucleotide, they will just inhibit the polymerization activity of the reverse transcriptase. So they do not produce or transcribe, I should say, the DNA molecule. So RNA DNA hybrid formation will be inhibited. Now after RNA DNA hybrid formation, RNase activity of the reverse transcriptase, that is the two activity, first polymerization and second RNase activity, will produce single stranded DNA. And now with the help of a special enzyme that we are not going to discuss here, that is not too much important, it will lead to the formation of double stranded DNA virus. This double stranded DNA virus then inserted inside the nuclear membrane, and inside the nuclear membrane, remember this thing, our host cell, membrane, okay, our host genetic material, and integrase, there is a protein that will cut it and add their viral genome inside. Okay, so now I hope that there will be another kind of inhibitor should be present that can inhibit integrase. Yes, obviously there are an enzyme, that is raltegravir drug present. This raltegravir drug is the integrase inhibitor. Okay, so after inhibition, after discussing about the inhibition, so what will going on, let's move to the normal case. So remember, inside that viral genome there is two LTR region present, okay, at the two end of the viral gene, and these LTR regions are nothing but cohesive end. Okay, and this cohesive end will possess a lock that is commonly known as, in the term of molecular biology, is promoter. Okay, and inside the promoter, if transcriptional factor binds, then the promoter will be activated and it will start transcription. Okay, that is the idea. So there are various transcriptional activators that activates LTR. Unfortunately, some host cells transcriptional activator activate LTR. That is not a good thing. That one of the example is nuclear factor kappa B. Okay, and but virus also develop its own key to unlock that lock. Okay, so we are going to discuss about those keys. So after the transcription has been started, structural genes are going to synthesized. Okay, so GAG, POL and ENB are the three structural genes. So GAG gene upon transcription will give you mRNA, and this mRNA will translate into a polyprotein first. Okay, remember one thing, that this polyprotein is not functional. Okay, so this polyprotein will be cut by protease and gives you three different protein. One is P24, which is the capsid making protein, and P17, which is the matrix making protein, and P7 is the nucleocapsid protein. Okay, similarly in POL, it first gives you a polyprotein. This polyprotein will be cleaved by protease and gives you our RIP, that is reverse transcriptase, integrase and protease. A similar case is going on in case of ENB2, but there is a one interesting factor that I want to mention, that is, in case of protease, in case of ENB, the protease is host protease. Okay, but in case of POL and GAG, the protease is viral protease. Can you guess what is the reason behind it? Yes, absolutely, you have predicted it right, that as GP41 and GP120 is not the own property of virus, right, it was the property of our cell membrane, right? So to activate them, normally our own protease incorporates. Okay, so very good. So we have discussed about our structural genes. Now let's think, as without the presence of protease, all the important and harmful protein of the virus cannot act. Is we have developed any kind of inhibitor to inhibit protease? Yes, obviously we have developed. So those protease names are, although I have forgotten unfortunately to incorporate this. Am I? Yes, I am forgot it. Okay, no problem. I will incorporate the name of protease in the description box. So still I will mention you here, that is ritonavir, sacubitril, darunavir. Okay, are the various kinds of virs that we are using as a inhibitor to inhibit protease. Okay, don't need to bother about that. I am going to attach the spelling of that drugs in our description box. Okay, so now the thing is, except the three gene from the same genetic material, again extra 6 regulated gene are transcribing. Now the question is why. Let's pause the video and start to think about